Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Scarlett Lowes from the Business Review, and I will be your host. It's our pleasure to have Bend Research, a division of Capsugel, with us today, who will be discussing accelerate your biologic process development using automated sampling. Today's guest speaker is Clint Pepper, PhD, and is a director. I'd like to welcome you to our webinar platform. You'll notice that this webinar is browser-based, so if you disconnect, please click on the link that you received via email to rejoin. You may move and resize the windows on the screen to customize your view. All of the icons along the bottom of your screen are the interactive widgets that we have on offer today, so please interact with them all throughout the session. In order to ask questions, you can send them in via the Q&A widget at the bottom of your screen, or please use the top left-hand box. We will allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. Please use the help widget if you experience any technical difficulties or require any more assistance. So without further ado, please allow me to welcome Clint.
Hi there, this is Clint Pepper. I want to thank you all for joining this webinar and talk to you today about how you can accelerate your biologic process development using automated sampling. Specifically, we're going to talk about your cell culture development and using the modular automated sampling technology, which we call the MAST platform. So some things we're going to talk about today. First thing is, what autosampling is and how it can accelerate your development. Then we're going to go through 3 different case studies. Then we'll talk a little bit about the MAST deployment reliability statistics. We'll be unveiling this product for commercialization soon, so I'm going to show you some of the current product development and commercialization items that we have. Talk to you about the modularity of the system, the MAST module configurations, and then wrap it up with a summary. So we'll jump right in. So first, discussion about our company. The MAST product line is out of Bend Research in Bend, Oregon. And Bend Research is a part of Dosage Solutions, which was recently formed in 2013, and that's a division of Capsugel. And you can see we have a pretty large global footprint. If you look over on the left side, Bend, Oregon, we're over there on the left, but we're now a global company throughout in every region of the world. And here in Bend, Oregon, we focus on developing new products and manufacture pharmaceuticals at the clinical and commercial scale. So we've been out talking as an engineering firm, as a problem-solving firm, helping companies solve their problems. And we've been hearing constantly that people need the ability to achieve their target quality attributes. And through research, we've really believe that the product quality attributes are a function of 2 things. One is the bioreactor environment. That's the media you put in, the temperature, the RPM, pH, those parameters. But it's also a matter of the cell state, like this the graph, this red and blue graph from Cabor of a Journal of Biotechnology. You can see that cells that look the same initially with regard to lactate output can diverge wildly because there's a different cell state. So understanding both the bioreactor environment and the cell state are very important to understanding your product quality attributes. So to develop true fundamental understanding of the bioreactor process, you need to measure and control these PQAs. And even though there are online sources now, you still need to be able to automatically collect samples from the bioreactor, and you need to be able to automatically analyze those samples. So here at Bend Research, we developed a system for collecting samples, and the core of the system is the sample pilot. And that's a system that collects whole broth samples from either fixed stainless steel reactors or development reactors or single-use bioreactors. And then we also developed a way to remove the cells from that solution to create a cell-free sample. So if you could collect whole broth samples and cell-free samples, then, like I said, you also have to analyze them. Collecting a sample itself is only half of it. So then you can send the whole broth samples to devices like the Nova, the high-res, BioHT, and other devices for measuring things like whole broth, cell density, viability, and other parameters. And then, to add to that, if you could take the cell-free samples and measure them for things like glycosylation patterns, amino acid concentrations, and titer, if you could do those things, then you could really have a control of your product quality attributes. And we'll go through this diagram here. Down on the lower left side, you have your bioreactor. You're pulling a sample with the sample pilot. It's a whole broth sample. And this the whole broth sample can be sent directly following the reddish arrows to your bioprocess analyzers. You can also send that whole broth sample to your cell removal system, where you remove the cells, and the cell-free sample goes to a liquid handler. We use Gilson a lot for that. You can do processing of the sample. You can do protein A on it, for example, or amino acid preparation, and then send that sample onto a downstream device like a UPLC or an LCMS. Now that you have this data, you can pull, following the gray arrows, you can take your online data combined with your at-line data and feed it into the green part of the diagram, which is your predictive models and your control algorithms, to ultimately control your bioreactor. So now that you can automatically sample and automatically analyze, it is truly possible to control your product quality attributes. So, how does this add value to your development program? So the very basic value from just auto-sampling is there is a cost savings over many years of being able to have samples taken automatically. What happens is you're able to direct your time to higher value activities, and you're not sampling on weekends and evenings. But really, the value comes from when you start to accelerate your development and scale up. If you are using something like an NIR or a Raman probe or a metabolite model, you can now take a lot of samples exactly and correlate that exactly to the spectra that you get from the Raman, for example, and train your model much faster. And then when you're starting to do runs, you can actually see your data in real time, so you'll know if your batches are having problems. And you can use a much more sophisticated design of experiments plan, which will reduce your experiments. Because you're going to be seeing your data in near real time, you can make decisions from your experiments, while the experiments are in process, instead of waiting weeks to get the data back from the experiment you just ran. The biggest value, though, is getting product quality attribute control, and that is where you can increase the productivity of the protein species, and then also reduce less desirable species and improve your safety profile. And that is where the significant savings comes in from implementing product quality attribute control. So, why is auto sampling so difficult? This is, as an engineering firm, we were hired by Pfizer 5 years ago to solve these auto sampling problem. And there was three main areas. The first one was maintaining sterility. A lot of issues with trying to auto samplers before were having frequent sterility problems. So we developed the sample pilot specifically to resolve this issue around sterility. And the idea with the sample pilot is we start from the bottom arrow. All of the sample pilots have a bioreactor isolation valve that's interlocked to prevent that valve from being opened at any time it shouldn't be. And then after every sample, when the sample is drawn, it's drawn into an aseptic zone, which has been previously sanitized and dried with alcohol. So that zone is a nice aseptic area to pull the sample into. It's at a lower pressure, so the sample's always flowing in. It creates a significantly improved sterility assurance profile. After every sample, we flush the contents and do a sanitant flush and hold throughout all the product contact lines, and then we blow that all dry with air afterwards. So you're getting your system back to its original state every time. The sample pilots are autoclave sterilized, either on the bioreactor or in a single-use bioreactor application. They can be connected to the bag afterwards. And the sanitant and purge gases that flow through are all passed through 0.2 micron rated filters. So that's maintaining sterility, but then also, delivering the sample. Well, that's difficult also. Sometimes the analytical devices are far away, and one of the issues that many of our clients have is they can't reliably get the sample to the destination. So we designed the sample pilot with a positive displacement pump directly inside the body of the sample pilot. And so we can use positive pressure to push the sample. We push a microbial sample 80 feet to the destination. We also have the ability to draw some sample if you're concerned about volume, and then use air at controlled low pressure to push that sample to its destination. Then the last problem that's seen frequently is clogging, and so this is a big problem, and we're constantly working through this. Is that cells accumulate in nooks and crannies of the pipes, and so the system has been designed very specifically to have valve seats that are very easy to clean behind. We take care to make sure that all the dimensions flowing in and out of the system, so there's no nooks and crannies or places to connect. But we still realize we have to flush with. That's one of the reasons we flush with alcohol. We flush all the contact components and all the sample lines every time with alcohol, and then blow them dry afterwards. So that's been a major step toward resolving clogging issues. So now I kind of give you an idea of what the MAST system is, the core of it. I'll jump into some case studies here. So this first slide just shows on the left an SP100 that's mounted on a microbial bioreactor. This is the SP100 is designed for stainless steel vessels that are steamed in place. And then the SP200, it shows it mounted on a bench scale bioreactor up in the white circle there. And that usually is mounted on a bioreactor, an autoclave with the bioreactor, or like I said, connected to a single-use bioreactor afterwards. So this first case study was automated protein A and amino acid preparation of MAST cell-free samples. So at this particular application, there are two SP100s on two different bioreactors in the field. Those samples are routed through what we call a sample navigator, which is like our traffic cop that directs the samples where they need to go. And out of the navigator, they're routed down through the cell removal system and into the Gilson GX271. So the Gilson has the ability to process the samples that are called out by the MAST system. So when we schedule a sample in the MAST system, you can tell the Gilson exactly what methods to run. For this particular application, the sample is split, and one part of the sample is done a protein A process on that sample, and then the other part of the sample is running an amino acid preparation on that sample. Currently, in this application, the next step we're working on is getting the integration of sending that sample to a Waters Patrol UPLC, and that's the part we're working on right now. So to give you a little bit of data on at least the protein A part of this, we wanted to test whether the Gilson protein A process was consistent with what we currently do in our lab. So we took 12 replicate samples, and we ran them on the GE ACTA versus our MAST-initiated Gilson protein A method. And so you can see that that all the results of these 12 replicate samples, 6 from each, is very consistent. So we're very happy with the way this automated protein A process is functioning. The second case study is some work we did here in our labs We have a measurement of the galactosylation in perfusion systems subjected to step changes in galactose. The idea here is we used a perfusion setup to collect data on dynamic cell response. And this gets back to what I talked about earlier about understanding the cell state. So first, we collected whole broth samples for automated analysis by the Nova BioProfile FLEX. And so you look at that upper red circle, you'll see there's two perfusion systems here side by side. And there's two SP200 on each one pulling direct whole broth samples. Then sample permeate after the permeate filter in the perfusion system. We pull that sample and direct that to the Gilson GX271 for a manual glycan analysis. And that's that lower red circle. And because we're doing a perfusion system, we're able to pull a cell-free sample directly, so we didn't need to use the cell removal system in this application. So that sample was sent to the GX271. In our lab, we have a database system that pings all of these instruments, pulls data from MAST, from the Nova, from the UPLC, and pulls that data all together. And then I'll show you the results from that. And the key with the MAST system is you can pull, when you need to pull a lot of samples, like when you make these step changes, you can get a lot of data right when you need it. So here's the concept. The idea was a perfusion step response, and the idea is that you'll see in the green lines represent the response back to the galactosylation when we did the step changes in galactose concentration. And so our perfusion system ran at a constant cell density, constant feed rate, and a constant volume. So here's the input. This is the galactose concentration in the blue lines here. So what the idea is that we made steady step changes in the galactose concentration, and we're waiting to see the output response. So the output response was the percent galactosylation. And you'll see the green line around 100 micromolar. Until the galactose reaches 100 micromolar, there wasn't a lot of control linkage between galactose and galactosylation. But after 100 micromolar, you started to see response from the cells increasing the percent galactosylation. And the key here is not only are you understanding the steady state, you're also understanding the cell response, getting back to the cell state. So if you see the galactose, initially, this was a step change. We added a bolus of galactose in the feed at a higher galactose concentration. It should have been a flat step change. But because you'll see this, there's a slope down, which means that the cells increase their galactose consumption. The other part is look at the galactosylation response. And here, not only do you get the new steady state value, but you get to see how the cells responded, how long it took them to change from change the rate of galactosylation. And this is key information that you have to have if you're making a control model. You have to understand the rate of change, not just the final steady state values. And one of the reasons that makes this possible is using a system like MAST to pull samples and do the automated analysis. Because you can see when we made the step change, we needed to collect a lot more data. So when you're waiting for cells to stabilize, maybe you can sample once a day, twice a day, but you might want to sample 6, 8 times a day when you are doing these step changes so that you can get the data that you need. And the only way to do this is with automated sampling, unless you want to be there all night on the weekends. The third case study is integration with the Nova BioProfile FLEX. The goal here is we provide samples directly to the Nova BioProfile FLEX, bypassing the RVM and the SPM. And we do that by mounting this external sample cell on the outside, and then we trigger the Nova to take the sample using the MAST system, and we can initiate the sample using all the parameters that are available in the Nova system. So here's one study in a pilot plant in Andover. We were sampling from 2 bioreactors for reg tox runs. And you can see in the lower right corner with the MAST data in red and the manual data in blue, the viability and the viable cell density were virtually identical between MAST samples and manually collected samples run on the Nova. Following that up with what we did in our labs, this is showing the viable cell density, viability, pH, and PO2 are very, very similar. MAST is in blue and manual is in red. Same is true for the metabolites, glutamine, glutamate, glucose, and lactate, virtually identical. And also the salts, ammonium, sodium, potassium, and calcium all showed very, very consistent results between MAST sampling and manual sampling. So, wanted to talk to you a little bit about where this MAST system has been deployed and some data on reliability. Currently, 7 companies are using the MAST system prototypes that we have put out in the field, and those are being used in 8 different facilities. There are 29 of the SP200s deployed and 7 of the SP100s deployed. Some of those are both in cell culture, microbial, and downstream applications. We've just recently put 5 cell removal systems out in the field, and we're working with those to define the best operational parameters for those. And we have deployed 7 of the Gilson GX271 liquid handlers for processing and analyzing samples. Now, we have taken thousands of MAST samples now. I think we might be at 10,000 samples without a sterility failure. So we've demonstrated the ability to maintain bioreactor sterility. We've used the system in cell culture, microbial, and downstream applications. And in our labs, if you ever get out to Bend, Oregon, you're welcome to come visit. You'll see our test stand where we're cycling these valves day and night. So I want to go over some of the recent product development efforts, kind of get a feel where we are. So the first thing is that we have created a commercial enclosures, and based on client input, the prototype systems that are in the field currently can control up to 8 sample pilots. This system can control 10, and it works with both the SP100 and SP200 units. We learned a lot over the last few years and incorporated those lessons learned from those prototype units into these systems, and they're completely modular and stackable. The SP200 has got some significant redesigns. We did a lot of things to increase sterility assurance and make a more robust design and just make it easier to use. You'll see on the left the original system that had a whole bunch of tubes and stuff sticking out of it. We were able to streamline that significantly into the version you see on the right-hand side. And this unit will be coming out in a few months. We did a lot of work to simplify and make more flexible the filter integration into the system for the sanitizing the sanitant and the air that comes in. We made it a smaller footprint with all the connections coming out the top because those bioreactors can get crowded, so it makes it a lot easier to connect and to keep the tubes out of your way because of all the other connections that go into your bioreactor. We improved and simplified the valve seat design, making it even easier to clean behind the valve seat. And there was an O-ring that we got out of the sterile side of the bioreactor isolation valve, increasing the sterility assurance profile. And so you can see we internalized all the pneumatic tubes and integrated the pneumatic umbilical into it, making it much easier to connect in the field. So the next part is a a lot of clients have used the sample pilots. When they're using them, when they're running a CIP, or if there's cleaning solution in their bioreactor, they can just take a sample to clean through the system. But if you don't do it that way, what we have is a cleaning station that we built, and can be designed to clean either ST100s or 200s. You just mount the sample pilot, and using its own pump and pneumatics, we're able to cycle cleaning solution and rinse out the system afterwards. So if this is something that you're interested in, let me know. So I wanted to go into a little bit more detail on the development of the cell removal system, so you can get an understanding of how that works. You see this the main picture here on the left, but the right, it has a door that opens, and then we have a removable cassette. So on the lower right-hand picture, you'll see the plexiglass sheet that contains the hollow fibers and the syringes that are used to process the samples. We've done quite a bit of work on the cell removal system over time. You see the picture on the left. That's not even the first prototype. That was like the third prototype, and that was a pneumatic-based system. And we found that we could not get the type of transmembrane pressure control we want to see across the hollow fiber filter with the pneumatic drivers. So we switched to a stepper motor-based system, which allows us to specifically tune the flow rates and control the transmembrane pressure much more specifically. The original system, you had to literally manually put in all the tubes and the hollow fiber cassettes, and so we made a quick change out filtration cassette that you can see in the picture there. We also created a system to added some complexity to allow it to have improved processing and cleaning operations and minimizing unwanted pressure spikes that happen when you are loading and operating the system, to reduce filter fouling and maintaining high level of operation. We also converted the system to use an independent PLC controller. We got a lot of input from clients. They might be interested in a standalone system someday where you could put 20 samples in a rack and have it process all of them for you in a separate independent way. So we made this have its own separate PLC controller so it could do that in the future. And we currently have 5 of these systems deployed out in the field. To give you an idea of how it functions, there's a very simplified diagram of how the system works. You see that the syringe on the left gets filled with a MAST sample, whole broth sample, and then that syringe is driven down at a targeted speed, and the syringe on the right comes up, and what happens is the sample gets pushed through the hollow fiber module. As the solution gets pushed back and forth through that module at a very controlled rate, we can pull out the cell-free permeate. This permeate accumulates in the syringe in the middle, and then is ultimately transferred to a downstream analytical device or liquid handling device. So this system, we have demonstrated the ability to separate cells from permeate for up to 40 samples before changing out the cassette. Big advantage with this system is that cell removal is external to the bioreactor. So if there is a problem with need to change the cartridge, or there's an issue with the filter, you can change the filter without risking the sterility of your bioreactor. The system has the full flexibility with respect to processing the sample and cleaning. We designed a system with a sequence that's modified, that's modifiable to change the way it operates to fit your exact needs. The protein recovery is being measured, and we're currently working on that. Protein recovery is dependent on the cell line. So what we found often is that you have to fine-tune the parameters, or even use a different kind of hollow fiber to get the kind of protein recovery, because we're finding that it's very cell line specific. And so we are experienced at working with people to find the right solution for their specific protein they're trying to recover. And as development continues, we're optimizing the hollow fibers we use, the cassette design, and other parameters. and we will continue to work with clients to get the highest protein recovery that's possible. Some other things we're working on is we are working with Roche Roche Diagnostics to integrate the two devices. One is the CDEX high-res. We have a system here in our facility. We have established a communication protocol with the high-res, and we have demonstrated the ability to initiate a sample at the high-res via remote triggering. We have a prototype that's in development that delivers neat or tenfold diluted samples to the high-res. And right now, as this project, we're waiting for a development partner to collaborate on an integrated solution to integrate the MAST to the high-res. So if anyone is interested in sending an automated sample to high-res, we have definitely demonstrated proof of concept and have some working prototypes. And then it would be a matter of making sure that it fits your exact needs to sample into your high-res and design a system that works exactly how you want it to. Same is true for the BioHT. We have developed communication protocols and demonstrated the ability to have a remote triggering of samples. And we also have a prototype that delivers samples to the BioHT. And again, we're waiting for a development partner to collaborate on integrated MAST to BioHT solution. One of the things we were very specific in designing the system. We wanted to make sure that the system could go all the way from the development scale all the way into GMP. We have designed the system to be GMP ready. We use Allen Bradley PLC hardware and industry-proven ignition HMI GUI software. The system is designed with using the industry standard accepted and traceable components. We have complete bills of materials. In other words, you can do complete IQ, OQ, and PQ on these items. The product contact materials, the materials that contact the inside of the bioreactor are either PEEK or 316L stainless. Sample contact materials are PEEK stainless, Radel fluoropolymers like PFA or FEP, and then the pump that drives the system is Buna. So we can put validation packages together upon request, depending on what your specific needs are, and we can put together software bolt-on modules that can be developed to satisfy your specific 21 CFR Part 11 compliance requirements. So, the M in MAST stands for modular, so I want to kind of go over the module configurations and how that works so you get an idea how the system can be constructed. You can go up to 10 sample sources, and you can send that to up to 4 analytical destinations. So the base level system is kind of the starter area. It's the smallest subunit you can put together. Starts with a master controller that can operate one sample pilot, either an SP100 or SP200, and send that sample to one destination, like the Gilson or a Nova or one other device. There's also in that system a computer with the ignition GUI software that has your basic scheduler, and that has a scheduling system for organizing all your samples to all your different destinations. It also has your database. It has your histogram showing all the activities that have occurred. It's a full, complete interface. It also has a sanitate and purge supply that's where your pump-based system that supplies your 70% IPA and the purge gases to blow everything out afterwards. So that's your basic fundamental system, and it can run one sample. And then it's quite a simple matter to add a sample navigator. Sample navigator controller has all the inputs and outputs with pneumatics to run up to 5 sample pilots and 1 sample navigator, which is like the traffic cop, which routes the samples to where they need to go. The master controller has the main PLC, and the sample navigator is a input-output slave to the master controller. So it's just an Ethernet connection and configuring the system in the GUI, and you're up and running with the expanded system. And like I said, the system is expandable up to 10, so you can add a total of 2 of these sample navigator controller modules that will allow you to pull up to 10 sample sources and route them to a destination. And the destination can be anything like the Gilson, or on this image you'll see the Nova. And these are things that we've done now several times. We have 8 of these systems out in the field, and we have a lot of experience doing this basic component. And if you know exactly what you're looking for, we can, you can call me and we can talk about what you need and coming up with a solution for that and generating a quote for that. We also have the capability as an engineering firm to help you design, different things for your specific applications. There's a lot of analytical devices. We're working on several projects right now connecting to the other unique types of analytical devices. And so this is often more of a development program where we take the sample and then we direct that sample to different destinations. We can pretty routinely send the samples to cell removal systems and the Nova and the Gilson, but there might be other devices like your specific, you have an Agilent HPLC or a Thermo Fisher mass spec. You might want samples directed there. So there's some development component to that, some engineering and software development work, and that would be a program where we could get together and develop a scope of work and talk through that. So in summary, we really focused on accelerating development, and now that there's automated sampling and automated analysis tools now allow the development of a biotechnology manufacturing process with true product quality attribute control capability. This is something that just over the last few years has changed pretty significantly in our industry. And what this allows is you to think differently about how you do your development. Whereas before you might have designed a large number of experiments to crank through a bunch of variables, now you can create different approaches. Like using in case study 1, for example, they are using the system to collect the samples, but also to do the automatic protein A and amino acid preparation. Just saving, in that just part alone, saving significant time and also creating a more reproducible manufacturing process. The second case study shows a completely new way here in our labs how we designed and operated an experiment using the perfusion system as a reaction cell where you can make step-based changes, not just on galactose, but say you could do different pHs or different other medias or other changes and watch the cell response. And you could set up an automated series of experiments that each run take 2 or 3 days to run in a perfusion system. So you could completely change your strategy for development using this type of approach. And then, the case study 3 shows just for routine operations how you can free up resource. When you're collecting samples and analyzing samples, you're not doing the more high-value work of taking your program to the next level. And I think that's where you'll see a lot of additional value is freeing up the resource to work on a higher priority development projects. So to kind of go through the advantages of the auto sampling, first we've created an aseptic zone to draw the sample into, and significantly increasing the sterility assurance. The positive displacement pump that's in the sample pilot is the key to consistently and reliably delivering samples. We don't have time-based operations or things like that. We are volume-based. Can be very consistent in delivering samples, even extended distances, and things that are quite viscous. This microbial sample we push is very thick, and getting that 80 feet to the Gilson is something that the sample pilot SP100 can do very reliably. The cell removal system that's external to the bioreactor, I think that's a very, very important aspect of that system. Our system is a closed system, so you can have high pressure flushing of all the lines, sanitizing the lines and drying, to reduce clogging and to clean out your system after every sample. And having a closed system makes it easier to run things under higher pressure and to return your system back to its original state after every sample. The system's totally modular. You can start small, and then we can bolt on these different navigators to add capacity. And we've really focused on making you're spending a lot of time setting up your bioreactor, I know. So we didn't want to make this as even more complicated. So we wanted to make it very fast to set up your bioreactor with as few connections as possible. And the system can be used seamlessly from development scale through to full GMP manufacturing. So what you learn at your development scale and your systems that you have developed can be, and your algorithms and your control strategies can be taken right into GMP using the exact same sampling equipment. And that's it. I'd like to thank you for your time, and I'm excited to hear your questions.
Thank you. It's now time for the question and answer section. Thank you to everyone who has already sent their questions in. Please continue to send your questions via the Q&A widget at the bottom of your screen or use the top left-hand box. We will try and get through as many questions as possible. However, if we do not have time to answer them all in this webinar, then Clint will get back to you at a later date. The first question is, what is the typical volume required per sample?
That's a good question. So the sample pilot SP200, which is used most, pulls sample in 5 milliliter increments. Generally, the first sample is drawn to clear out the dip tube, and that's usually pushed to waste with purge gas. And then usually you can pull a 5 or 10 or 15 mil sample afterwards to send to your destination. If volume is not a concern, you can use more and push liquid with liquid. The system's flexible in the way that you can set up the schedule sequence. So usually the smallest volume you'd pull is about 15 mils, but you can pull more if you want.
Great. And the next question, can you change out the CRS cassette during the middle of a run?
Yes, the system is specifically designed to be able to change out that cassette, and so if there is an issue with the hollow fiber module or in your very long perfusion run, for example, you can change that out and get your system back to its original starting position without risking bioreactor sterility in any way.
Thank you. And the next question, do you have to replace all of the sample lines every run?
No, that's a very specific thing that we designed the system to be user-friendly and quick to install. And so the majority of the sample lines are in a FEP or other fluoropolymer tubing that is cleaned between each use, so that whole setup through the navigator and all of that can be fixed in place. And then part of our routine maintenance program is to replace that, say, annually. We come out and put new tubing in to replace that. But it's kept in place run to run and cleaned between each sample.
Thank you. And the next question we have, did you look at other liquid handlers besides Gilson?
Oh yeah, so we did. We explored many different options, and we found Gilson to be the most reliable. Definitely great customer service, It gets back to us quickly, and well-priced unit, and a whole lot of flexibility. I have yet to find something that this liquid handler can't do. There's other systems that can transfer, process a bunch of samples at once, like a 96-well plate system. But what we're really processing is one sample at a time, and so this system really fit what we were trying to do.
Thank you. And unfortunately, this will be the last question now. What is the delivery time for one of these systems?
So delivery time, I can answer that two ways. Delivery time, for the whole system. That's depending on if it's a straightforward system, then you're probably looking at 8 to 12 weeks. We have a very engaged process where we initiate the clients with a questionnaire. We do a site visit, walk the plant, and we set up routine meetings, usually bi-weekly meetings, where we go through all the things that are required. There's a lot of discussions with IT, for example, to integrate these systems into your research environment. And we work a lot of those through, and then we come on site, install, and train. And so, if possible, we also like the people to come out to our facility to do a test run of the system and get some training also. It takes about 10. So it takes usually about 10 to 12 weeks to deliver a system. If there's more development involved, where we are integrating something special, that can be longer, and we can do the project in phases. So we're very flexible here.
Great. So sadly, that's all we have time for in today's session. If we did not get round to answering your question, then Clint will get back to you at a later date. So that just leaves me to thank Clint for what was a great presentation, and thanks to Bend Research for sponsoring this session. The attendees, you will receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. Thank you once again, and I hope you all have a nice day.